James Temple quoted in The Future of Food that in a 1932 essay about the future, Winston Churchill predicted, "50 years hence we shall escape the absurdity of growing a whole chicken in order to eat the breast or wing by growing these parts separately under a suitable medium." He was close in his prediction. 68 years later, NASA and Harvard cooperated together in an attempt to create a new form of food for astronauts. They weren't the only ones in search of a new form of nutrition. Over the next 4 decades, demand for meat products is projected to double, and meeting the demand for the increase in meat cannot be done with current production methods. Green and animal rights activists have long pointed out that the modern food industry is environmentally unsustainable and cruel. Vegetarians often think it is unethical to cause suffering to animals, and this turns away people from meat produced from them. In vitro meat, or SHMEAT, is one solution NASA and Harvard indirectly created for this problem. In vitro meat is created through the use of a few cells taken from parts of an animal, such as prenatal sheep, cow shoulder muscles, fish, or frog skeletal muscles, that are then grown in a medium. These cells are considered to be stem cells. Once a sufficient amount is produced, thin layers are then minced and mixed with animal fat, also lab-grown, to form a ball of meat. To reach the required amount, it takes upwards of 20,000 strands to produce a 140-gram patty, and thousands of layers of cells to create a ball of meat. For the creation of a single burger, cells must be taken from a newborn calf's neck. These are myosatellite cells, a type of stem cell. These cells are then cultured in a growth medium containing fetal calf serum or a derivative of cow blood. The medium must contain vitamins, lipids, and amino acids needed for the cells to grow and multiply. The cells are cultured in incubators between 36 and 39 degrees Celsius and must be moved to new flasks periodically due to contact inhibition, meaning the cells are unable to grow on top of each other. The cells are then taken away from the growth medium and poured onto a bit of gel, where they are starved of nutrients and forced to differentiate into skeletal muscles. However, these cells are immature and lack protein, so a second differentiation step is required. This can be achieved through either mechanical or electrical stimulation. Mechanical can be achieved by pulling the culture over a post and creating a slight vacuum beneath. On the other hand, electrical stimulation is created by placing 2 electrodes in the culture and running a charge through the cells. The cells will then continue to grow and differentiate into myoblasts, single muscle cells, that in turn create myotubes that eventually organize into muscle fibers. The result is a tiny strip of meat half an inch long and 1/25 of an inch in diameter. These strips are combined with thousands of others, as well as fat cells, to create a burger that tastes and resembles real meat. Not every organization that has been working on stem cell food has been able to achieve a taste resembling that of the true meat version, but several have come close, like in the case of Utrecht University. NASA and Harvard were only able to achieve a small amount of taste, so their food was relatively bland. They produced fish fillets that they extracted from fish shells, which yielded little flavor, but this was just the beginning of stem cell food research. New Harvest has performed experimentation of its own in the growing field, and they have produced products such as sausage, different types of hamburgers, and chicken nuggets. New Harvest deduced that any basic, boneless, processed meat can be produced through various manipulation methods of the stem cells, and any flavor can eventually be produced through the artificial incorporation of fatty acids, seasonings, and proteins. New Harvest director, Jason Matheny, stated, "A hamburger in vitro can have the fatty acid composition of salmon." This shows that stem cell meat can be turned into essentially anything. Harvest was able to produce lower quality versions of what was essentially steak from the prenatal sheep cells. James Temple quoted in The Future of Food, "Independent of other advances, in vitro technology will never produce filet mignon or a pork chop. The mouthfeel in traditional cuts has as much to do with intermixed blood vessels and fat as the muscle itself. Adding vascular tissues to engineered tissue is certainly a goal of scientists, but is a long way away."
Matheny notes that the technology, given 5 to 10 years, will exist, allowing for the proper addition of various fatty acids, proteins, seasonings, vascular structures, and other flavor additions, along with various manipulations of the meat to create different textures. Mark Post, a leading scientist in the field of in vitro meat, believes that new food technology can be commercialized in a decade or 2. In vitro meats should then reach the supermarket and allow for new types of meat such as ground beef and the hot dog to be created, as well as meats with different nutritional content. Thanks to the fact that stem cell food is more or less a blank slate that can be manipulated into anything, the nutritional benefits will eventually vary greatly. As previously mentioned, the director of New Harvest stated that the fatty acid composition of the produced burgers can resemble that of a salmon. Jackie Arnett mentions in her article, The Green Plate Special, that stem cell burgers can also mimic the fatty acid profile of an avocado, leading to healthier options for humans. In essence, the nutritional value of in vitro meat is dependent upon what the company decides to put into the meat. This also means that the FDA will have to heavily monitor the food to make sure nothing harmful is placed into the meat. While they do not work specifically with food, StemTech, considered to be a leader in the stem cell nutrition industry, has discovered and utilizes various types of supplements in their products, showing that effective efforts to introduce supplements and nutrients into stem cells are already underway. New Harvest is experimenting with their own methods of incorporating nutrients. One way they do this is by attaching the in vitro meat to a sponge-like scaffold, and then soaking the cells with nutrients, and then mechanically stretching the in vitro meat to increase the size of the meat cells and their protein content. By encouraging the consumption of in vitro meat, humans can avoid the accidental consumption of diseased meat that can occur through poor farming habits, such as feeding the cattle the remains of other cattle that carry harmful pathogens. It also helps avoid the issue brought about by meat substitutes and falsification of the meat people are consuming. An example would be horse meat in the place of beef, which is being incorrectly advertised as beef. Besides these promising qualities of in vitro meat, there are still many items people disagree with. Currently, the process costs over $330,000 to create a single product. However, as more research is done in the field and in vitro meat becomes a more commercialized product, the price will drop drastically. Others believe that the creation of meat in a lab is unnatural and this sort of science should be avoided, but the final product is almost identical to a real burger, and is still made up of the same cells and will have a similar taste. Oftentimes, different cultures view and divide animals differently. While in America, dogs are viewed as pets and cows as food, different parts of the world make different distinctions between which animals are and are not food. In Southeast Asia, dogs and cats are consumed with cows. Over time, people's views change as to what is and is not food, and eventually in vitro meat will most likely become a common food item. Several current issues should be resolved through the use of in vitro meat. First, this new process provides a more humane process of meat production. Animal rights activists have long been opposed to the cruelty of the current methods used to procure meat. The use of stem cells to produce meat would create an almost animal-free process in the production of meat. This would also provide vegetarians with a good protein source, since no animal will be harmed in the production of in vitro meats. Another issue is the amount of resources used for animal farming. Nearly one-third of the Earth's landmass is used for animal farming, and much of the grains produced in agriculture are fed to livestock. The production of in vitro meat will mean that less land will need to be devoted to livestock, and more of the grains produced on farms can be used by humans. As the demand for meat rises beyond what the current methods can produce, the importance of stem cell-based meats will rise. For the time being, stem cells will remain under experimentation in laboratory conditions while more research is done on them. An affordable, commercially available version that people will want to consume is still several decades away, but in vitro meat is currently our best hope for solving our future food-related crises. While Winston Churchill may have had the time a little off, he predicted the future of the food industry very accurately.